Hello, in this video we will look at the muscles. Basically, we will understand the type of muscle and their physiology on the basis of their structure and their types. So, for example, a muscle is used for the contraction and relaxation for locomotion as well as the movement and locomotion. So, the skeletal muscle, cardiac muscle, and smooth muscle is we can categorized, and that is skeletal muscle are present. It is the part of the peripheral body part which that is attached with the bone with tendon. So this tendon is attached, that is the non-elastic part of the muscle, while on the other hand the cardiac muscle, which that is cardiac muscle, is related to the heart. So this is the heart muscle which that is different as compared to other, and the smooth muscle is the spindle like structure which that is part of the digestive system and blood vessels. So here is the skeletal muscle which that is contain muscle fibers. These muscle fibers contain more than one nuclei. So on the other hand, if we will discuss about the cardiac muscle, which that is also same nuclei, which that is more than one nuclei, but important thing is that they contain a intercalated disc or gap junction. This gap junction is very important, which that provide the action potential forward throughout the cells, throughout the tissue, for complete contraction of the whole heart, whole muscles. Why? Because it is non-skeletal muscle, so bones are not present, so it means it's contract through a cell to cell, so it is impulse creation. So here we will discuss about the property of smooth, cardiac and skeletal. So if we will discuss about the appearance, the unstriped and smooth, and cardiac is the irregular, and skeletal is the regular stripes. While on the other hand, the cell shape is a spindle, branched, and spindle or cylindrical in the case of skeletal muscle. And the number of nuclei is one per cell, smooth, and many per cell, both cardiac and other. So the speed of contraction is a slow by state of nervous system control, sympathetic and parasympathetic for the smooth muscle, while the spontaneous movement due to the cardiac and the nervous system, which that is the skeletal, which that is controlled by the voluntary, but the cardiac is the involuntary. So here we will discuss about the skeletal muscle, their anatomical aspect. So here is the compact bone which that is attached with the tendon, and the muscle bundles are attached. These contain a fascicles, and these fascicles contain a inside is known as the muscle fibers. And this muscle fiber, the fascicle, is also contain blood vessel, which that provide the oxygen and accept the carbon dioxide. So in this way, the affinity of the loss of oxygen from hemoglobin is more less in the near to the muscle, so the dissociation will be easy. So here is the muscle fibers. This muscle fiber is basically inside is the muscle fiber, and that's contain a inside the fascicles which are surrounded by the muscle fibers, and this muscle fibers thick is 10 to 100 micrometer, and this is basically inside is the sarcolemma. So the sarcolemma is inside is of the we will discuss later, so you should understand about that. Here is not the sarcolemma, but inside the sarcolemma we will discuss, and it is the part of the muscle fiber used for the storage of the glycogen and hemoglobin for providing the oxygen, and the glycogen is important for the providing sugar quickly by the dissociation or glycogenolysis. So the important thing is that the muscle is used for the movement and sport and movement, as well as the storage in the form of glycogen and hemoglobin. So that is important things. So important thing is that vitamin B12 is the important for providing the hemoglobin. So that is why the vitamin B12 deficiency will lead to muscle weakness. So here important things that next is we will discuss about inside is the sarcolemma containing inside is the uh, inside is known as the sa- inside is the sarcolemma and it's surrounded by the muscle myofiber which that contain a myofibril, and this myofiber is contained basically inside the interstitious fluid. So the sarcolemma is surrounded by the myofibrils, and there are many myofibrils are present, which that is also endoplasmic reticulum or sarcoplasmic reticulum, which that is store the calcium, and calcium storage sac containing sarcoplasmic reticulum are present in the surrounded by the sarcolemma, and inside is known as the sarcomere, and this sarcomere is contain many myofibrils contain many sarcomeres. Sarcomere is the unit of the contraction. So here is the nerves also present. This nerve is the motor neurons. These motor neurons is used for the neuromuscular junction for activation of the muscle contraction through a brain signal by a sensory neuron afferent to efferent receptor to effector. So this is the effector, and it is synaptic knob are present, and this synaptic cleft is present, and the mitochondria is used for the providing energy. And acetylcholine is the neurotransmitter which that is bind with the acetylcholine neurotransmitter receptor, and that's sodium potassium exchange will be occur in the post synaptic membrane, and this post synaptic membrane basically the synaptic knob are present, which that is a positive inside and the negative outside, which that will be depolarization and again repolarization to contract the muscle by a changing of the calcium ion in the cytoplasm. So in this way we can think about that. This is the neuromuscular junction. So let's begin to understand with the again we will discuss about. So first of all we will discuss about the mechanism of the reflex arc in the effector to effector neurons. For example, the sensory to motor neuron, how that work. So we will discuss here. Here is the spinal cord, that is the part of the central nervous system. This is the part of central nervous system used for the reflex arc, and this is without going or going to the brain for a third order neuron and second order neuron, or without going to the brain signal, the sensory neuron will be sense the stimulus, for example fire, pressure, touch, or something. So that will be integrate into the gray matter inside of the spinal cord, and out is the white matter. Butterfly structure is known as the gray matter. And the peripheral nervous system efferent motor neuron will be emit that signal for the effector, and this efferent motor neurons and will produce the effect to contract this hand, for example this contract this muscle for prevention of the danger, for example the needle will be uh, uh, effect that. So this is the sensory and this is the motor neurons. Okay, here we will discuss about the next is we will discuss about the myofibril, and this myofibril is basically contain a many sarcomeres unit. For example, the 9 is basically study shows that the 9, but here I am not drawing the 9, but you can understand as well. But here is the diameter of the myofibrils is a 1 to 2 nanometer, and this contain a basically actin and myosin filament. This filament is basically there are many filaments, which that is the part of the contraction, and this is the contracting unit which contain a sarcomere. This is the sarcomere. So the sarcomere, what is that, and why we will say that the sarcomere? We will discuss. The mere means the smaller part. It means the minimum part. That's contractile unit of muscle. So this is the surrounded by the sarcolemma, which that is black color shows that, and inside here is the myosin head and myosin filament and actin filament. So the myosin filament is the thick filament, while the actin filament is the thin filament. So remember about that. Here I am trying to understand the contraction of the muscle. So first of all, we will say that the contraction of sarcomere. And what is that? Here is the actin, black color, and actin contain a troponin as well as a tubulin, while the myosin is the thick filament which contain myosin heads. And this is the contracted state of the muscle. When this contraction, after the contraction will be relaxation, require energy, and in this way the hydrolyzation of the actin and myosin bridge, and this bridge will be hydrolyzed to relax this muscle. Without ATP, it will not relax. So it means the rigor mortis will be occur after death, and that muscle will be cramp and irreversible movement. So that is why. And here is the actin filament, and here is the myosin filament, and it is the relaxed state. So here let's begin to understand the neuromuscular junction, and that will be the part of the nervous muscle. So here we are covering the whole process of the muscle contraction and the types of the muscle. So here is the skeletal muscle, which that is used after the sensory neuron sense the signal of the hand, for example, and this transmit to the spinal cord of the gray matter from the dorsal horn of the gray matter of the uh, uh, yes, and ventral ganglion. So this will be transmit the signal toward the spinal cord, and after the integration, the neuromuscular junction, this neuromuscular junction basically will be synapse, and this synapse is basically known as the neuromuscular junction. After this neuromuscular junction, basically muscle will be contract due to the neurotransmitter acetylcholine. This is the neurotransmitter. So let's begin to understand with the synaptic knob, and this is the axon terminal, and this axon terminal of the neuron, that type of neuron is the motor neuron, because the sensory neuron sense and motor neuron contract of the muscle. So the effector to the effector process, and the interneuron is the inter between the effector and effector. So here is the synaptic knob, which that is the end terminal of the axon, which contain a knob-like structure, and the neuromuscular junction. So here is the calcium will move inside after the nerve impulse, which that stimulate the calcium move inside near to the knob, end of beginning of the knob, and the calcium will move inside, and here is the choline acetyl transferase enzyme, which that is transfer the acetyl CoA as well as the choline, which that is combined together and to give a new structure. This new structure is known as the acetylcholine. And this acetylcholine will be exocytosed, and this is the neurotransmitter will be bind with the acetylcholine receptor, and after the binding with the acetylcholine receptor to allow the sodium move inside, so the influx of the sodium into the muscle cell. So first of all, I want to say about that. Here is the voltage gated potassium channel, which that is the potassium will move out, and after the voltage gated potassium channel, which that is 3 to 4, and this 3 to 4, and this is used for the depolarization, and after the depolarization will be calcium will be also stimulate the exocytosis of the acetylcholine neurotransmitter. After the binding with the new acetylcholine receptor, the sodium will move inside, and after the complete process, the choline will be separate from the acetyl-CoA, and this choline will be back move into the neuron from a high affinity choline transporter channel, which that is allowed to again movement and repair by the acetyl-CoA binding with the choline esterase enzyme, and that will be again exocytosis. So the continuously movement of the muscle is the important things, because the choline will be not end product. So here is the voltage gated sodium channel will more enter, and that impulse will create the more sodium inside. And after this more sodium inside, the calcium will want to cover that depolarization process. So the calcium will move inside through a um, T tubule. So here is the T tubule, transverse tubule. So the here is the transverse tubule, and RYR1 is the acceptor which that accept the calcium, and the sarcoplasmic reticulum will be allowed to efflux. So that's efflux of the channel from the efflux of the calcium will lead to bind with the troponin, yes, and this troponin binding with the release with the contraction. So this contraction after the releasing of the calcium, ATP is used to hydrolyze this head, which that is myosin head containing pair of the head. So this is the relaxation. After the contraction of the muscle hydrolyze, and the sodium will be again back to the cytoplasm. And here is we will discuss about the cardiac muscle contraction. And what is that? Basically is the pacemaker are present. Here is the pacemaker, which that is a sinoatrial node, which that transmit the signal to the atrioventricular node or AV node. So here is a pacemaker which that transmit the signal throughout the cells. And this depolarization, the calcium will be moved to another cells, another muscles. So that means the depolarization will be occur, and this depolarization forward move, and that is the here is the intercalated disc or gap junction. This gap junction is important for the calcium move to another cell. So that is the cardiac muscle system. So it will create the action potential. So let's begin to understand basically the action potential with a bigger picture to understand here. So the pacemaker which contain after the stimulation of the pacemaker through a nerve impulse from the sympathetic division or parasympathetic division, which that is both is able to contract far fast or low or slow contraction. So here is the pacemaker which that contain a calcium, and this calcium is the move to myocytes. Basically is the excitation contraction coupling. Basically here is the myocytes will be accept this calcium, and more calcium move through by excitation and contraction coupling. So this the action potential will begin through a more calcium move inside. It means the sodium will move more, like a previous diagram, just a neuromuscular junction. So that is why here is the gap junction are present. This gap junction is contain a channels which that is move, and here is the sarcoplasmic reticulum contain a calcium. This calcium will be move more outside, efflux of the calcium. So the in this way the calcium will move forward to contract whole cells. So that is why the whole muscle contraction will be occur throughout the heart for ejection of the blood. So here is the sarcoplasmic reticulum, which that is efflux of the calcium, and the calcium move out or pump out into the cytoplasm or sarcoplasm, and that contraction will begin by the binding of the calcium with the troponin. So the troponin is here, and you can see that is the troponin which that is bind with the calcium, and after this stimulate the myosin head to bind to contract and hydrolyze through ATP to convert ADP. So that is why this is the function of the muscle contraction and the muscle anatomy. So I hope you make sense. Next video we will discuss about the smooth muscle contraction and their sympathetic and parasympathetic division, and we will also understand the stimulation for digestion. So that is here is the end of the video. So thanks for watching. Please make sure to subscribe.